Hello everyone, my name is Miss Sonu, working as an assistant professor in IIMT College of Pharmacy. Today, I will discuss here about the urinary system. Let's start with the course content. It contains introduction of urinary system, anatomy and physiology of urinary system, kidney, nephron, physiology of urinary formation, urine formation, renin-angiotensin system, maturation reflex, clearance test. Now let's start with the introduction of urinary system. Urinary system of the body is formed by the organs responsible for converting the excess fluid and other substance into the urine, its filtration and its excretion from the body. These organs include kidney, ureter, urinary bladder, and urethra. Urine is formed by the kidney, then sent to the urinary bladder for storage and finally excreted through urethra. Urine excretes excess of minerals, vitamins, and blood cells from the body. Urinary system, along with other system of the body, maintains homeostasis. The kidney play a major role in homeostasis as they maintain the acid base and water and salt balance in the blood. Now, uh, anatomy and physiology of the urinary system. The urinary system play a major role in the urine formation and urine elimination. Normally, urine in person is about 1 to 1.5 liter per day. Urinary system consists of a pair of kidney, a pair of ureter, urinary bladder, urethra. Now discuss about the kidney. Kidney are the reddish brown bean-shaped structure covered by the connective tissue capsule situated between the level of last thoracis and third lumbar vertebra close to the dorsal inner wall of the abdominal cavity. Each kidney of adult human measure about 10 to 12 centimeter in length and 5 to 7 centimeter in breadth, 2 to 3 centimeter in thickness with an average weight of 120 to 170 gram. There is a depression on the medial border of the kidney called hilum through which renal artery, renal veins, nerves and ureter pass. The component of the kidney are arranged into two layers, that is cortex. Outer layer is cortex and inner layer is medulla. Outer cortex, it is a dark and granular appearance and it extends in between the medullary pyramid as renal column called column of Bertani, and the inner medulla consists about to 8 to 18 medullary or Malpighian's pyramids. Further, it continues in renal pelvis by forming of 2 to 3 minor calyx and about 8 major calyx. This is the diagram of kidney. This is internal diagram of the kidney and urinary system diagram. Now we will discuss about nephron. So nephron is a structural and functional unit of the kidney. Each kidney consists approx 1 to 1.3 million nephron. Nephron is divided into two parts. First is renal corpuscles, yeah, malpighians corpuscles. It is situated near the cortex region of the kidney and help in the filtration of the blood. It is formed by two portions. First is glomerulus. Glomerulus is a tuft of capillaries enclosed by the Bowman capsule. It consists of glomerular capillaries interposed between afferent arterioles on one end and efferent arterioles on the other end. Second is Bowman capsule. It covers the glomerular capillaries and continue with the tubular part. Next is renal tubules. It is continuation part of the Bowman capsule on the basis of structural and functional basis. It again divided into three parts. First is proximal convoluted tubule, second is loop of Henle, third is distal convoluted tubule, and last is collecting duct. Basically, nephrons are of two types. First is cortical, also called superficial nephron. Renal corpuscles situated near the outer cortex with a short length of loop of Henle, and it helps in the urine formation. Peritubular capillaries present all around the cortical nephron. Second is juxtamedullary nephron. Renal corpuscles situated in inner cortex near medulla with long length of loop of Henle and help in urine formation and mainly in the urine concentration. Vas recta capillaries present all around the juxtamedullary nephron. Next is physiology of urine formation. When blood passes through the glomerular capillaries, the plasma is filtered by the filtering membrane into Bowman capsule. Urine formation includes three processes. First is glomerular filtration, second is tubular reabsorption, third is tubular secretion. So glomerular filtration, it is the process through which the capillaries blood going to filter through the filtering membrane. It is also called as ultrafiltration because the minute particles also filtered through it except the plasma protein and the blood cells. The filtered fluid is called glomerular filtrate. Second is glomerular filtration rate. So it is defined as the total amount of filtrate formed in all the nephrons of both the kidney in a given unit of time. The normal glomerular filtration rate is 125 ml per minute or about 180 ml per minute. Next is tubular reabsorption. Tubular reabsorption, it is the process through which the needed wanted substances are absorbed by the renal tubules and transported into the back to the blood. When the filtrate pass through the tubular part, the large amount of water, electrolyte, and other substance reabsorb and resulting qualitative and quantitative changes occur. These are the sites of reabsorptions. First is proximal convoluted tubule. About 85% of the substance reabsorbed by the PCT, that is proximal convoluted tubule, glucose, amino acids, sodium, potassium, calcium, bicarbonate, chloride, phosphate, urea, uric acid, and water are the major substances reabsorbed by the PCT. Second is loop of Henle. The sodium and the calcium ions are main substance reabsorbed from the loop of Henle. Third is distal convoluted tubule. Sodium, calcium, bicarbonate and water are reabsorbed from DCT. Then collecting duct, the condition reabsorb happen in the collecting duct because water reabsorb in the presence of antidiuretic hormone, that is vasopressin, and sodium reabsorb in the presence of aldosterone hormone. Tubular secretion, it is the process by which the substance are transported from blood into the renal tubule. It is also called the renal excretion. The site of excretion, the potassium is excreted actively by sodium potassium pump in proximal and distal convoluted tubule and collecting duct. Ammonia is excreted in the proximal convoluted tubule. Hydrogen ions are excreted in the proximal and distal convoluted tubule. Maximum hydrogen ion excretion occur in proximal tubule. Urea is excreted in the loop of Fennel. Now let's discuss about the renin-angiotensin system. So renin-angiotensin-aldosterone system, it is essential for the regulating the blood volume, systemic vascular resistance. Juxtaglomerular apparatus, it is a specialized modification situated near the glomerulus of each nephron. It is formed by three different structures, that is macula densa, extraglomerular mesangial cells, extraglomerular cells. So the renin-angiotensin aldosterone system activate under the influence of stimuli. These are the stimuli: low blood, when the low blood pressure, low ECF volume, sympathetic stimulation, low plasma sodium. These conditions stimulate the juxtaglomerular apparatus, which release the renin enzyme. The renin enzyme convert the angiotensinogen into the angiotensin first, then uh, angiotensin first convert into the angiotensin second in the presence of angiotensin convertase enzyme, which are released from the lungs. Then angiotensin second convert into the angiotensin third in the presence of angiotensinase. Then further angiotensin third convert into the angiotensin fourth. The angiotensin second that help in regulate the glomerular filtration rate, increases blood pressure. It helps in increases water intake, increases ADH secretion, increases CRH and ACTH secretion. Angio third increase the aldosterone secretion, and it also cause vasoconstriction. Now, maturation. Maturation, it is the process by which the urine is eliminated from the urinary bladder. It is a reflex process. Urinary bladder, it is a triangular hollow organ in which ureter open. It is formed by the smooth muscles called dextrosor muscle. It is open into urethra via internal urethral sphincter. Urethra Urethral structure and function different in male and female. In both, two urethral sphincters present in urinary tract. First is internal urethral sphincter, second is external urethral sphincters. Urinary bladder and the internal urethral sphincters are supplied by the sympathetic and parasympathetic division of autonomic nervous system. The external sphincter is supplied by the somatic nerves fibers. Now let's discuss about the mm, maturation reflex. Maturation reflex initiated uh, by the stimulation of the stress receptors. Stress receptors situated on the wall of the urinary bladder and urethra. When about 300 to 400 ml of urine is collected in the urinary bladder, then sensory impulse reach to the spinal cord through the sensory fibers of pelvic nerves, and motor impulse produced in the bladder and internal urethral sphincter. Motor impulse cause the contraction in detrusor muscles and relaxation in internal sphincter, so that urine enter the urethra from the urinary bladder. Once the urine enter in urethra, then urethral stress receptor sense the change and send sensory impulse to the spinal cord via pelvic nerve. Now the impulses generated from the spinal center inhibit the pudendal nerve, so the external sphincter relax and maturation occur. During maturation, the flow of urine is facilitated by the increase in abdominal pressure due to the voluntary contraction of the abdominal muscle. Next, clearance test or renal function test. And these are the properties of urine. First is volume. Normally the volume of urine filtration is 1 to 1.5 liter per day. The slightly acidic pH about 4 to 5, 4.5 to 6. Specific gravity is 1.0102 to 1.025. Osmolarity is 1200 milliosmo per liter. Examination of urine. Routine examination of urine or urinalysis is a group of diagnostic tests performed on the basis of urine. First is physical examination, second is microscopic examination, third is chemical examination. Physical examination, we observe first color, abdominal coloration of the urine. Then straw color is due to severe causes like jaundice, hematuria, hemoglobinuria. Appearance, turbidity appear in both physiological and pathological condition. Volume, increase in urine volume indicate increase in protein catabolism and renal disorders such as chronic renal failure, diabetes insipidus, and glycosuria. Specific gravity, it is low in diabetes insipidus and high in diabetes mellitus, acute renal failure, and excess medication. pH is useful in determining the metabolic or respiratory acidosis and alkalosis. Microscopic examination, uh, red blood cells. The presence of red blood cells in urine indicate the glomerular disease such as glomerulonephritis. White blood cells number increase in acute glomerulonephritis, infection of urinary tract, vagina, or cervix. Crystals, abnormal crystals such as crystal of cysteine, tyrosine appear in liver diseases. Bacteria grow appear during the infection condition. Chemical examination, we in the chemical examination we ab- see we observe glucose normal 180 mg per dl, protein normal 30 mg per day, ketone bodies urobilinogen 1 to 3.5 mg per day, bile salt, bilirubin, blood. Plasma clearance is defined as the amount of plasma that is cleared of a substance in a given unit of time. It is also known as renal clearance. It is based on the Fick principle, that is C equal to UV divided by P. U is the concentration of substance in urine, V is the volume of urine flow, and P is the concentration of substance in plasma. So that's all about of the urinary system. Thank you. Thank you everyone.